SARS-CoV-2 Omicron virus evades human immune system and stays infectious. New findings from a research published in Science offer light on the SARS-CoV-2 Omicron variant's ability to evade antibodies and its ability to maintain strong connections with the receptor, which are two of the most important aspects behind the virus's remarkable ability to spread. The Omicron form of SARS-CoV-2, first reported in November 2021, was soon recognized as a serious mutation of concern by the World Health Organization (WHO). Currently, even among those who have received 2 doses of the vaccine, this particular variation is spreading, posing a serious threat to the public's health. Vaccination was based on the knowledge that SARS-CoV-2 enters cells through the angiotensin-converting enzyme 2, ACE2, receptor recognized by its spike glycoprotein. Omicron variant spike protein, on the other hand, is the most mutated virus variant so far. It has 37 mutations. Since the Omicron variation has already spread, it is imperative that we understand how these mutations affect ACE2 receptor binding and antibody evasion, as well as those that may evolve in the future. A team from the University of British Columbia's Department of Biochemistry and Molecular Biology and the BC Centre for Disease Control Public Health Laboratory in Vancouver, Canada, worked together on this project. Cryogenic electron microscopy, cryo-EM, was used to examine the Omicron spike protein structure in unprecedented depth in a new article published in the prestigious journal Science. Researchers have also done surface plasmon resonance studies to get a better idea of how Omicron spike glycoprotein mutations affect the binding affinity of the ACE2 receptor on human cells. They then compared the apparent binding affinities of spike glycoproteins from wild-type and delta strains. SARS-CoV-2 spike glycoprotein antibody neutralizing monoclonal antibodies were also used to assess the effect on monoclonal antibody escape of Omicron RBD and NTD mutations. It was shown that R493, S496, and R498 of Omicron variant spike glycoprotein with cryo-ER in association with human ACE2 were responsible for novel bridges and hydrogen bonding between the 2 proteins. Because of these interactions, additional Omicron mutations known to lower ACE2 binding affinity, such as K417N, seem to be compensated, leading to identical biochemical ACE2 binding affinities in both Delta and Omicron variants. Furthermore, the cryo-EM structure of the spike glycoprotein ACE2 receptor complex reveals a structural basis for this behavior. Other ACE2 binding effectiveness seems to be regained through interactions involving new mutations in the Omicron version at residues 493, 496, 498, and 501. Like aforementioned K417N, the changes in Omicron variant spike glycoprotein, which are unusually prevalent, likely to be the cause of a very wide antibody escape compared to previously emerging and circulating variants of SARS-CoV-2. For individuals previously infected with Alpha and Delta versions, there was a significant decrease in neutralization power against the Omicron variation compared to the Delta variant, according to a new research. Omicron's resistance to neutralization seems to be stronger than previously thought, based on this research and other recent studies. Prior infection or immunization is required for this to occur, and it is more prominent than other variations of concern that have developed during the coronavirus illness 2019 pandemic. In any event, the Omicron variety seems to have evolved to balance the ability to evade neutralization with the ability to effectively bind to the ACE2 receptor. Final but not least, the rapid spread of the Omicron variation has substantial biological and public health consequences due to the increased antibody evasion and the maintenance of robust contacts at the ACE2 interface.